As we start to delve into this whole idea of the way cells divide, let's just do a quick review of the cell cycle and make sure you know where we're going and where we've been. Remember that generally speaking, a cell doesn't spend much time dividing at all. It spends about 10% of its life. Some cells, in fact, never divide. And we'll talk about, we'll talk about that later. But the bottom line is, here, G1 is where most of your cell's activities are going to occur. Then the cell starts to get ready for replication. It grows a little bit more in G2, the G standing for gap, but we want to spend our time right now in this section right here. This section, which is going to be called mitosis and cytokinesis. Now, immediately, the vocabulary starts coming out like asteroids out of the sky, all right? And yes, biology does have a lot of vocabulary. And yes, the word mitosis is important, and the word cytokinesis is important, but I haven't even written them down yet. So am I a derelict in my teaching here? No, absolutely not. We're going to use those words plenty. But what I want you to understand at this point is the cellular events that are going to happen right here in this stage called mitosis. And then we're going to talk about the events that happen in the cytokinesis stage. All of this stuff, the interphase, the phase when the cell is not actively dividing, we've spent a lot of time and we'll spend a lot more time on that as we study cell physiology. But now we want to talk about right there, that M phase. What's going to happen when a cell divides? Later, there'll be plenty of time for the names of the phases. But right now, let's see what happens. Okay, well, let's quickly review what's up with chromosomes. What's a chromosome all about? Remember, the chromosomes are about packing of DNA. You only pack DNA into a chromosome when you're going to do mitosis. Mitosis. Better write that down. Sorry, guys. Vocabulary. Can't speak without vocabulary. All right. So in this mitotic division, or in this mitosis, we're going to form chromosomes. And chromosomes end up looking like something like this. And let's talk about what makes something a chromosome. Well, remember, it has its DNA, and its DNA is packed, and there's a structure on that chromosome. All of this would be one molecule of DNA all the way down, all the way down, all the way down, all the way down, all the way down. Yet in the center of the chromosome, there's this kind of pinched in section, and that's called the centromere. And within the centromere is going to be a very important structure, okay? And we'll see why it's so important in a second. And that is going to be called the kinetochore within the centromere. The kinetochore. Kinetic energy. Starting to think it might have something to do with motion, right? Well, if you're thinking that, you're absolutely correct. All righty. So what mitosis is about, number one, is forming chromosomes. And just so I can show you, you can form chromosomes in three different ways. The centromere doesn't always have to be in the center, okay? This one in particular has its chromosome in the center, but some can be off-center, and indeed, some can be at the very top of the chromosome. So anyway, these three chromosomes, this metacentric chromosome, this acrocentric chromosome, and this telocentric chromosome, all have their centromeres in different places, but that's not as important as what's going to happen here. What's going to happen is going to evolve the kinetochore. Now, once again, I want you to think about the concept of mitosis rather than memorizing phases. What's your job? What's your goal? If these are the chromosomes in their doubled form, what is it you want to do with these chromosomes? If you're a cell, remember the purpose of mitosis, to form an identical copy. And therefore, your role is to get these chromosomes so that you have half of the, so that you can actually half them, because they're doubled, and then split them. That's all it is. We double, we split. We double, we split. We double, we split. That's all you do your whole life, okay, with your cells. Well, that doesn't sound so hard. And really, the whole idea of mitosis, phases aside, is more a concept of what happens in your cells and your cytoskeleton and in your cytosol, then what happens to your chromosomes? Because that's very important. I want to talk to you a little bit about how chromosomes move. There's the magic of mitosis, chromosomal movement. One of the things you're going to notice, as I just said, all right, we got our chromosomes doubled, now we got to split them. Well, in order to split them, you got to move them. You know, you got to take a chromosome like this and somehow get it to go like that. And you got to somehow organize these before you can make a move. And that is where spindle fibers come in. Spindle fibers are all about movement. Now, you know about spindle fibers a little bit. When we talked about, if you may want to link back and read about tubulin, okay? Because spindle fibers are made out of tubulin. Tubulin is a protein, okay, that makes up microtubules. So spindle fibers, therefore, are microtubules. You remember that the cytoskeleton consists of microfilaments and microtubules. I know you know that. Well, here's the thing. As we enter mitosis, what's going to happen is the microtubules of the cytoskeleton are going to break down. Why? Well, here's the thing. You from these cytoskeleton microtubules, you are going to make these things called spindle fibers. Why? Because they're going to be made out of the same material. Using the British spelling of fibers there, I guess it's okay. Alrighty. And so the point here is that you have a cytoskeleton in your cell. That cytoskeleton is there during G and S and G2, but as G2 progresses, it starts to break down. Why? You're going to use the raw materials of this microtubules, the tubulin, to make fibers across the cell called spindle fibers. You'll see those and what they do. Well, I'll tell you right now. What's the purpose of the spindle fibers? Well, one of the things that I said has to happen is that your chromosomes have to get organized. They somehow have to get set up. And let's just take this randomly occurring cell, this chromosome laying in the cell right now. And you know what? Let's just say I want to somehow get these chromosomes to organize and line up before I split the cell. Well, right here, denoted by this paperclip, is going to be the kinetochore. Okay? So what you're going to see is that as the microtubules grow, they're going to hook to a kinetochore. We'll put it all in sequence when we learn the phases. But they're going to hook. And then what starts to happen is, remember what tubulin is? Tubulin is a protein that sits on in a circle around in a circle that makes this long tubular arrangement. In fact, take a look at the box right now. You can see that whole tubulin arrangement. What happens is, apparently these have an ability to shorten. But you know what? If this is a microtubule, and this is where it's hooked to, and you know what? We might be able to even make this just a little more dramatic by putting it under an orange background. Huh? Okay. If this is a microtubule and it's hooked over here, it doesn't shorten at its anchor site. It turns out it shortens at the kinetochore. So we have a microtubule hooked to the kinetochore, and there's an involvement of a motor molecule there. So like a Pac-Man, what's going to happen is this. This is going to shorten here by digestion, but the paperclip, if you will, is going to eat its way along there, and it's going to cause chromosomal movement. What does that look like to someone watching this? Well, it looks something like this. Here's a microtubule hooked to a kinetochore, and literally, it looks like it's being reeled in like a fishing line. But then, another microtubule hooks to the kinetochore on the other side of the chromosome, and it starts to reel back. Now, these are hooked in the center of the cell, and then this one reels, and then this one reels, and then this one reels. And what you eventually get is that doubled chromosome in the center of the cell. And you do that with all of your chromosomes. Do you know how many chromosomes humans have? We have 46 chromosomes. All 46 of your chromosomes will be lined up. How? And what do we call this? Names can come later. But now that you have them lined up, guys, it's easy. Since they're all attached by their kinetochores to microtubules, the next step is once again the motor molecules, and 1, 2, 3. You've split your cell into 2 groups of chromosomes. See, that's the principle. We're going to put some names on it soon, but that's all mitosis is about. Doubling chromosomes and then splitting them with the help of microtubules.